In interphase, the cell starts to get ready for mitosis, where the DNA gets replicated. In prophase, the centrioles start to move apart. They then move to the opposite poles, and the nucleolus and the nuclear membrane have almost disappeared. In metaphase, the doubled chromosomes line up at the mid-cell. In anaphase, the centromes split and half move to one pole and the other half to move to the other pole. The nuclear membrane then forms around the separated chromosomes. In interphase, DNA strands thicken into chromosomes. Homologous or like chromosomes begin to approach each other. Homologous chromosomes pair to form bivalents. The centrioles divide and move to opposite poles of the cell. In metaphase 1, the tetrads attached to the spindle fibers at their centromeres line up at mid-cell. In early anaphase 1, the tetrads separate and the paired chromatids move along to the spindle to the respective centrioles. In late anaphase 1, the chromatids have almost reached the spindle poles. The cell membranes begins to constrict. In telophase 1, nuclear membranes enclose the separated chromatids. The cell membrane completes its construction. The first mitotic division ends. There are now two cells, each with the same number of chromatids as the parent cell. Prophase 2 begins. In the second mitotic division, homologous chromosomes do not duplicate but merely separate. In metaphase 2, the chromatids line up at the mid-cell. The centrioles and asters are at the pole. A spindle has formed. In anaphase 2, they are now separated chromatids approach their respective poles. The cell membrane begins to constrict. And telophase 2 has been completed. There are now 4 cells, each with half the number of chromosomes of the parent cells.